to Mr. DeSale's podcast on how to properly process and graph your data. So I'm going to go through this one. I'm going to be using the program Numbers, a Mac program. So then I need to show you how to do this, and I will have a separate podcast on how to do this in Excel. So here we go. Now I've set up some data, and there's certain things that I set up that are common mistakes that happen. And I will explain those as we go. But the first thing I need to do is I want to process this data. So I made up an experiment where there was oxygen produced, probably a catalase type of thing. And I have my units here. Now, one of the common mistakes is when we get to like degrees Celsius, like I have here, a computer program doesn't know how to graph degree C. It's not a value. So it only graphs values. So when I go to my graphing data, I am going to remove these, and I'm going to just put that as a title and just keep it as a value. You can keep it in your main data table with your degrees C if you like. It's not a huge deal. Okay. So that's the first major common mistake that people make. And they wonder, why is my graph turning out weird? Well, it doesn't know what degree C is. Okay? So when in biology, and especially IB biology, they require you to do 5 trials. So we got our 5 trials, and we have actually 5 different independent variables. So these are the temperatures. So we do not graph individual trials. That is not how you display your data. If you were to do that, it would turn out really confusing. So if I just selected all of this and said, oh yes, let's graph these. Well, take a look at that. What does it tell you? Remember, your graph tells the story of your data. I look at this, I don't have any idea what it's trying to say. So what we do is we always graph the means, okay? So the average of your trials. So what we're going to do is we're going to go over here to the mean, and then we're going to put in an equation. So we start with an equal sign, and that gets a function, and we'll start typing in average. So right there it comes up. So I select average, and then I select the values that need to get averaged, and then I hit the little green check. Bingo. All right. So I can do that for the rest of this. So equal AV average, and then I select that, and I go to green. How nice. I think you're getting the idea here. Because why we need to do the averages is because, you know, in biology, data varies. Living things are not all uniformly exactly the same. So the more trials we have, the better and more accurate we can determine our data is. And that's the bottom line. Let's see here, one more here. Equals AV. There may be a shortcut to doing this. I haven't figured it out on Numbers. I know it on Excel. All right, so we have means. Okay, so we have averages here. And what we also need to do is we need to calculate standard deviation. Now, standard deviation is how precise your data is. And this is what we're going to use as a source for your error bars. In biology, standard deviation is primarily used. So what we're going to do is we're going to do another equation, equal STDEV. Okay, so we got STDEV, and then we select just the trials. We don't select the mean. And we hit OK. And we're going to do that here, where is it? STD. Okay. Now do not do the mean. That would throw things off quite a bit. All right. Okay, equals STDV. So this will tell us how accurate. So the bigger the value, it means there's more variation in your data. And this has to be showing up on your graph. Now, if you notice that all of these values have been rounded to different degrees of decimal places. So I typically like to round everything off to either tenths or hundredths just to keep it all consistent, because it's kind of all over the map. You look at this and we got, oh, this is major places. So what we're going to do is select all of our number values here. Then we're going to go into over here, and there's the format, and so we got table, cell, text, arrange. We're going to go into the cell, and right here, our data format. So this tells us how many decimals. So I'm going to go to hundredths, because I think that's pretty cool. All right, so we go up 0, that's even, 1 decimal place, 2 decimal place. And see how nice and uniform that all is? Sweet. Okay, now what we need to do is we cannot graph just from this data table, because over here we're going to be graphing the mean and we're going to be graphing standard deviation, and these are formulas. These aren't values yet. You see the value, but underneath there is a formula. So what we have to do is detach the value from the formula and cut and paste it here. So in numbers, we go and we select that, and we do Command Copy, Command C. Now, we're going to put this down here, but we're going to do a special kind of paste. So we're going to paste formula results. That's just the values. So they're going to be there, so there's no underlying values. And we're going to do the same thing for standard deviation. So Command C, get over here, and paste formula values. All right, so now we have processed this data, and because this data is like a various trend, we're not comparing distinctive different independent variables. This is the amount of processing we need to do. But if you are comparing completely different conditions, you're going to have to do a t-test. And we'll cover t-tests in a separate podcast. But this is the processing we need to do. All right, so first thing we need to do is we need to start setting up a chart. Okay? So let's take a look at what we're going to do. All right, so what we're going to do is we're going to select our temperature, which is our independent variable, and the mean of our oxygen collected. Okay, and then we go up to charts. And in biology, either bar graphs or scatter plot graphs seem to work best. I'm going to use a scatter plot graph. So that's this one down here. Okay. All right, so now I have a graph that shows me, wow, there is a general increase in trend as temperature increases, but I'm missing quite a few things. All right, I'm missing a title to my graph, I'm missing X and Y axis, and I'm missing error bars. So let's put in the error bars first here, okay? So I'm just going to click on one data point. See how they're all highlighted now? And what we're going to do is we're going to do y-axis error bars. They fit most things in biology best. And we're going to do both positive and negative. So now they come up with just a standardized error bars, but we're not done there. That's not what we want. We don't want a fixed value. We want custom values. Okay, so now we have to put in values that say the positive values are the values above the point, above the data point, and the negative values are below. That's what makes a total error bar. So we're going to use, like I said before, our standard deviation. So I go down and I erase that 10, and then I go up into my data and I highlight my standard deviations. And then make sure you hit the green check. That's really important. So now I go into the negative and I erase those and I highlight the exact same standard deviations. All right, now look at that. All the bars are very unique to the data sets. See how we have different standard deviation values and they are reflected here on the graph. Pretty cool. All right, now some teachers like to have you put in a trend line. I don't think that it really enforces much of anything. I think you can clearly see that there's an increase. Now what we're going to do next is we're going to put in a title. So here, this little dot up here is for a legend. For this case, we don't really need to worry about a legend, but we do want a title. And so you can obviously overwrite this. Mean oxygen production. That's fine. You can have a little form of the research question too if you like. All right, but I'm still, if you look at this, and there's no thing right over here for y and x axis. So we can go here, and then we can go to, okay, let's see here. Actually, I'm going to go do an easier way. Okay, so this has some different, okay, so we got x-axis name right here. So then you can write this in as degrees Celsius. Or you can just, if you want to, I think it'll work, where you can just cut and paste something from here. Copy. Let's try it. There we go. Degrees Celsius, and I might as well cut and copy this right here. Copy. And then when we go to this here, we gotta go to Y value, axis name. All right. So now we have X and Y axis labeled. We have a title. We have data points. We have an error bar. If you would like to, there are certain ways you can format these things. If you want to have some people to make their data stand out a little better, they do a little background fill. And so you can, you know, put in a little bit like that. Don't get too fancy. Don't put in really dark colors. Make them really light. And then when you want to, and then you can, what you can do is just Command copy when you select on the graph. And then if you want to, here's a Word document, totally different software, and then Command V or paste. And there you have your graph. Pretty sweet. So I hope this was helpful in how to process and graph in numbers. So anyhow, that's it for now. If you have any other questions, bring them to class and I'll be happy to sit down with you. Bye-bye.